My microbiology students, now that we've reviewed the gram stain, acid-fast stain, capsule stain, endospore stain, and wet mount that you have learned to do in lab, let's move on to Table 3-4. This is on page 223 of your lab manual. If you notice, you have an array of bacteria in this column here. We are crossing out this bacteria for this assignment. And notice we may be using Pseudomonas aeruginosa instead of Pseudomonas putida. This makes it easier on you. Notice for this gram stain column, it tells whether the organism is gram positive and stains purple or gram negative and stains pink. For the cell morphology column, it tells you what shape the cell is. Is it a rod? Is it an irregular rod? Is it an ovoid cocci that may be difficult to tell whether it's a rod or a circle? It is a cocci circle. What is the cell arrangement? Are they found in only single cells, tetrads, chains, pairs, or irregular clusters? Your acid-fast stain can be either positive or negative. If it's acid-fast negative, it'll appear blue. If it's acid-fast positive, it will appear reddish purple. With your motility column, this shows you whether or not the organism is motile and moves around while alive. If it's motility positive, it moves around while it's living. If it's motility negative, well, it is non-motile and it's not going to go anywhere. It is stationary. For the capsule stain column, this will show you whether or not your microorganism produces a capsule or not. And if it's capsule negative, there will be no white halo around the cell, indicating there is no capsule and it is capsule negative. If it is capsule positive, it will have a nice bright white halo around the dark little cell, showing that there is a capsule present in this capsule positive organism. For your endospore stain column, they will appear either positive or negative. If it is negative, the endospore will not be present and there will be no malachite green in your lab slide. If it is endospore positive, you will see a malachite green endospore somewhere within your bacteria. Notice, most people can see these are clearly positive or negative, but they do get confused very easily on what that actually means and what that represents in your bacteria. So I've made this chart for you that spells it out very blatantly what it means. If you notice, next to the cell arrangement, I've drawn little images to signal what palisade may look like, what single cells may look like, what some chains or pairs would look like, what some tetrads would look like. Notice they're kind of boxy with 4 of them right next to each other at 90-degree angles, or whether they're in clusters, or whether they're in irregular clusters that kind of look like grapes. So, if you notice, this one has a unique arrangement. That can help you identify it. Also, please note, whenever it looks like this, it is endospore negative, and there is no malachite green present in the slide. So, if you would like to take a screenshot at this time, it may benefit you during lab.